You're listening to the Animal Free Labcast, the podcast that puts humans, not animals, at the heart of medical research. Hello everyone, welcome to the Animal Free Labcast. I am back again with my co-host, Dr. Gemma Davies. Hey Gemma.
Hi Carla, how are you?
I'm good, thank you. How are you?
I'm good, thanks too.
So what's happening in today's episode?
Well, we're going to have a chat about our science conference that we went to the other day. It's also World Parkinson's Day on the day that this episode comes out, on the 11th of April. And we've also got a special guest who has just finished his project researching Parkinson's disease, Professor Jens Schwamborn from the University of Luxembourg.
Brilliant. So we'll be hearing from Jens in a little while. Yeah. So before we get stuck into all of that, Gemma, what about the poll results from last week?
Yeah, so the question was, do you agree with the APPG's purpose, which is to build cross-party support for replacing animal experiments in medical research? Yes or no? And
the results?
The results are, we had a very positive response with almost every vote, 270 votes for yes, and just one vote for no.
Very uncontroversial.
Yeah, it is.
Well, let's see if we can get a slightly more controversial one and get a bit of debate going next week. I think we've got an idea.
A good one for you
that,
this week.
Yeah.
So what's grabbed your attention this week, Carla?
Oh my goodness, it's been such a busy couple of weeks. It's been really good.
We haven't stopped.
So the week before last, we were in New York, so me and you, and also over 100 other people, all the scientists at the forefront of the animal-free innovation revolution, as we like to call it. So well over 100 people at our conference that we were co-hosting with the Humane Research Trust and Replacing Animal Research, so two of our kind of friends. We decided to get together and put on the conference together this year and invited all of the scientists that we all fund together to come along and just discuss the work, the groundbreaking work that everybody's doing. We had a whole load of sponsors come and help us to sort of support putting the conference on as well. Organ-on-a-chip companies, animal-free antibody companies. A really fancy company that makes 3D bioprinters, which is very cool to see. This is like a 3D printer, but they kind of print what cells? Is it cells?
Yes.
Yeah. Very cool.
Yeah, it's amazing.
We'll try and get a photo of that for you because it's very cool. We also had keynote speeches from our very own Professor Jeff Pilkington, who's the chair of our board of trustees, Dr. Malcolm Wilkinson, also on the board of trustees, and Dr. Dharminder Singh from CN Bio, which is one of the organ-on-a-chip companies that's doing really well, doing a lot of work over in America, but based here in Cambridge. And then we also gave out this year's Pioneer Awards. So Outstanding Scientist Award went to Dr. Dania Movia for her groundbreaking liver research. And we're going to get Dania on and do a podcast episode with her later in the year. We also gave our Early Career Scientist Award this year to Dr. Laura Sinclair of the Exeter ARC for the amazing research that she's doing on aging. And then we had our Supporters' Choice Award, so your award, which went to Professor Chris Denning for his groundbreaking animal-free heart research. So it was really lovely to be able to present those awards to them in person. And if you follow along on the Animal-Free Lab Stack or on our social media, you'll see photos of us giving them the awards soon, as soon as we see them. I think that should be any time. So that was the week before last. And then last week, another hugely busy week, but really exciting, really positive. So I went to Westminster with Isabel, our public affairs director, to talk to politicians who are helping to make Herbie's Law a reality. We had some really good meetings. And then something that we can't say much about yet, but we were back in London, weren't we, Gemma?
We were.
Working on a very exciting thing that we're doing for...
So exciting.
World Animal Free Research Day, which is on the 27th of May. So it's coming up soon. secret squirrel.
Watch this space.
Yeah, can't say much about it, but we will do soon.
It'll be worth the wait. Very exciting.
What about you, Gemma? What have you, I mean, obviously you've been involved in most of that. What else have you been thinking about?
Yeah, we've been doing so much. Well, I've been thinking about, as it's World Parkinson's Day today, which is the 11th of April, this episode comes out, I thought it might be useful to have a bit of a chat about Parkinson's disease and see where we are in terms of research and treatments. Because I think it's one of those things, one of those diseases that people have heard of and might have a bit of experience with, either themselves or through a loved one that's had it, but maybe you might not know too much about what it actually is and why we so urgently need new treatments.
Yes, please tell us more about it, Gemma.
So Parkinson's disease is the world's second most common age-related degenerative brain disease. So Alzheimer's disease is the most common and Parkinson's is second. And it's estimated to affect about 1% of the world's total population over the age of 60. And that's about 10 million people worldwide.
That's a lot of people, isn't it?
It's so many. And the prevalence has doubled over the past 25 years.
Is it?
So it's huge and it's growing quickly. In the UK, around 2 people are diagnosed with Parkinson's every hour.
Oh,
and
goodness.
That works out about 153,000 people are currently living with Parkinson's disease. But the problem is, as we are generally living longer, this number is estimated to grow by about 20% in the next 5 years. And it's just going to continue to rise.
Is that why we're seeing the rise in numbers? Because people are living longer?
Yeah, because as you live longer, the symptoms get worse gradually.
Okay, they get more developed. Okay.
Yeah, exactly. And we don't actually have any cures for Parkinson's disease. So as these symptoms progress over time, when people are generally diagnosed around the age of 60 or around that age, you can live for many years after a diagnosis with, a normal life. You might have some symptoms that can change how you live your life and the things that you can do, but you can live a normal amount of time, but it's generally the kind of complications that come with it that eventually lead to death, sadly.
So we know that Parkinson's disease affects the brain, but what's actually happening to the brain? Why do we get it?
Well, we don't really know, to be honest. We know that it is a progressive age-related disorder that primarily affects the central nervous system, so that's the brain and the spinal cord. Mm-hmm. And the main problems that you have with it is it causes issues with movement. Yeah. So the most well-known characteristic symptom is a tremor in the hand, and it often starts in one hand, but it can spread to the other limbs, but also different movement problems like slowness, rigidity, and posture and balance problems, the kind of common movement problems. But it can also have wider impacts on things like mental health, problems with memory and thinking, depression and anxiety and poor sleep. And it can also lead to things like effects on other bodily functions and even cause you to lose your sense of smell. But all of these develop slowly over time and get worse as it progresses. I
don't think, Gemma, until we've been talking about it, that I was really aware of the mental health kind of issues that come with Parkinson's, aside from, the kind of diagnosis and the kind of upset that must cause, but that actually there's something happening in the brain that is causing depression and anxiety.
Exactly. And I think because it is that kind of non-specific symptom, it can make it quite difficult to actually get the diagnosis because, that could be related to something else. It could be confused, or just missed.
Yeah. So people might not know that is a symptom of Parkinson's.
Yeah, exactly. So all of these symptoms are due to a loss of the nerve cells in the brain called neurons, in a very specific part of the brain called the substantia nigra, which is in the midbrain. And the midbrain is right at the top of the brain stem, and it kind of links the brain to the spinal cord. So it's a really specific area. So
it's right in the middle of your head.
That's it, right deep at the bottom of the brain. And these neurons from the midbrain project their kind of long extensions into a different part of the brain called the striatum, where they secrete a neurotransmitter chemical called dopamine. And this acts as the messenger.
Dopamine's the happy hormone, right?
That's right. And that's kind of why the loss of dopamine that we see in Parkinson's disease is linked to things like depression, because it's kind of not transmitting the messages properly.
Right.
But dopamine is also really important to help you coordinate your bodily movement. So that's why when we lose the dopamine producing neurons, we also lose the ability to move as normal. So basically what happens in Parkinson's disease is these nerve cells die or become damaged and the amount of dopamine they produce is reduced. And then, of course, you kind of lose the ability to move normally, and the different symptoms that come with it. But the problem is we don't really know what causes the loss of these cells or who is at most at risk of this happening. We know that men are more likely to have Parkinson's than women. We know that age is one of the biggest risk factors. And like I said, the average age of diagnosis is about 60. And there are some links to environmental factors like pesticides or air pollution exposure. But aside from that, we don't really know. There are some genetic factors that can predispose you to developing Parkinson's, but we're not really sure why or how. But unlike other neurodegenerative diseases like Alzheimer's disease, for example, it's quite rare for Parkinson's disease to be inherited. So even if one or both of your parents have Parkinson's, it doesn't necessarily mean that you will.
So you said it's usually quite a slow onset disease. So does that mean it's usually diagnosed quite late? You know, people have lived with Parkinson's for quite a while without really knowing it.
That's right. And like I said, you know, some of the symptoms are so generic, you know, you might not actually realise that anything is really wrong. But the main symptoms of Parkinson's generally only start to develop when about half of the nerve cell activity has already been lost. So this, you know, makes it difficult to diagnose at an early stage. But also once it is diagnosed, it's already a lot of damage has been done. Yeah. And at the moment, there isn't really any specific tests that can conclusively show that you have Parkinson's disease. Yeah, it's kind of diagnosed with a mixture of mental or physical tests that can, you know, give clues towards a diagnosis, but the symptoms can be confused with other things or kind of written off.
So doctors just sort of doing their best guess, really, of like, well, we think you've probably got it given how you're presenting.
Yeah.
Right. And then what happens if you do manage to get a diagnosis? Like, what are the treatments available?
Well, at the moment, there is no cure for Parkinson's disease. There are some treatments that can help relieve the symptoms and maintain quality of life. Okay. And once you have had a diagnosis, things will be put in place to help. So things like physiotherapy to help with movement issues and things like that. But some medications can help with symptoms, you know, things like the tremor by replacing or substituting the dopamine that's been lost in the brain, or to help stop it being broken down. There's drugs that work in different ways that might work better for different people, but they do come with some quite nasty side effects and they don't always work, unfortunately. But the other thing is they don't stop the progression of the disease, they can just manage the symptoms. So it is going to continue to progress even if you're on these treatments.
So we really do desperately need more and better research being done, and presumably part of the problem is we've been researching in animals rather than looking at the human situation, so we've been modelling the wrong thing. But it sounds like, you know, it's you know, there's a real critical need for better understanding and then treatments for patients.
There is, you know, we don't understand what's happening in the first place to be able to develop treatment. So we really do need to, you know, take it back to what's happening in the human brain and go from there.
That's just heartbreaking, isn't it? Thinking about all the research that just hasn't really led anywhere.
It is. It's really upsetting. And like I said, it affects so many people around the world. This really is quite urgent.
Yeah, we need to do better.
One of the things you might have heard about recently is there is a more invasive treatment option that you can have if these medications aren't working for you or if they have stopped working where they were working before. And this is called deep brain stimulation. And this is where a pulse generator, similar to a pacemaker, is put under your skin in the chest or the stomach area, which is connected to some fine wires that are inserted very specifically into that midbrain area, and a tiny electric current is produced by the pulse generator to stimulate that part of the brain. And this can help control some of the symptoms of people that have, you know, the really bad movement problems. But again, it's not going to stop the progression of the disease. And you might have heard about this technology in the news over the past few years in relation to Neuralink, which is Elon Musk's company that are developing brain-computer interfaces for this kind of thing for lots of different reasons. But one of them is to help treat neurological diseases like Parkinson's.
That sounds quite good, Gemma. That sounds like, you know, like quite a scary procedure to have maybe, but like if it could help, that sounds good.
Yeah, well, unfortunately, they haven't had much luck with it so far. And the project itself has found itself kind of surrounded by controversy because they are testing these devices in animals, things like sheep and monkeys, and they have been reported for potentially violating the US Animal Welfare Act.
Oh my goodness.
And
the US Animal Welfare Act, I mean, it's not, my understanding is it's not as good as what we've got in the UK, and we've been talking about what we've got in the UK being very far from ideal as it is.
Exactly. Yeah. They don't even keep track of the numbers of mice, rats, birds and fish, which are the most commonly used animals. They don't actually report on how many of those have been used, which is likely to be very big numbers.
That's the big numbers. Yeah. Okay.
So yeah, it is different in the US.
Well, Neuralink need to be doing human-specific disease, I think, is the quite simple message.
It is.
If we want to understand diseases in humans, let's start with humans.
Yeah, exactly. And, Elon Musk has pretty much unlimited money and resources to throw at this, but he's still not getting the results that we need for Parkinson's patients. And a large part of that is because they're testing it in animals rather than human-specific research, so we can understand what's actually happening in patients.
So, Gemma, why are people continuing to use animals when we're not making progress?
Well, as with most research, that we've talked about before, it is the gold standard. It's the way it's always been done. Animals, are always thought to be the best way to understand these kind of diseases. But diseases like Parkinson's and Alzheimer's, where we need new treatments so desperately, are uniquely human diseases. Animals don't naturally develop diseases like Parkinson's. So the only way that we can even begin to see these kind of diseases in animals is either to genetically modify or surgically alter these animals, but it doesn't mean that they fully show all the symptoms that humans show, and also we can't study them long term because animals don't live as long as humans. So this really important long-term progression of the disease, which we need to be able to study, we can't do that using animals.
Well, and also studying symptoms isn't studying the disease, it's studying symptoms, which are, just an effect of the disease. So that doesn't sound very ideal. No. Okay, well, presumably there are some researchers who are looking at this in a human-specific way.
Yeah, absolutely. We're funding some incredible projects on neurological diseases, Parkinson's and Alzheimer's disease. And you can read about these in more detail on our website. But I can also now introduce our brilliant guest for today, who has just finished his recent project with us, who was studying Parkinson's disease in a completely animal-free way. So let's welcome Professor Jens Schwamborn, who is Professor of Cellular and Developmental Biology at the University of Luxembourg. And he's also co-founder and CEO of Organotherapeutics. Welcome, Jens. Thank you for joining us on the podcast.
Thanks a lot for having me here. So my name is Jens Schwamborn. I have kind of two affiliations. On the one hand, I'm professor for cell biology at the University of Luxembourg, and I co-founded a small biotech company also in Luxembourg named Organotherapeutics a couple of years ago. And what we are doing is we are trying to develop in vitro, so cell culture models for Parkinson's disease that are patient specific. So everything that we do starts actually with a sample from a patient, and then we use that to create these cell culture models for that particular patient with a focus on Parkinson's.
So why have you chosen to focus on Parkinson's disease?
So when I set up my own lab already 20 years by now, more or less ago, my idea was I wanted to obviously work on the brain because that was during all my studies the focus that I had, and I thought I needed a disease model. And at that stage, I was naive enough to think that Parkinson's is quite simple because dopaminergic neurons in the midbrain degenerate, so it's one cell type in one brain location. Obviously, we know now this is way oversimplified, so it's way more complex. But still, I got stuck with the topic, so to speak.
Well, it's a fascinating topic, and obviously it affects so many people, all over the world. So it's a really important area to work in.
What would you say are some of the kind of key barriers to getting better treatments for patients, Jens?
Particularly for Parkinson's, I think our biggest problem here is Parkinson's is not a single disease. It's more like a syndrome. So it's kind of an umbrella term that probably summarizes a lot of diseases that just lead to a similar clinical endpoint. So we would actually need drug discovery that is already very well patient stratified. So that looks at small, well-defined subgroups of patients. So we probably don't need one medication. We probably need 20, 25 targeting different populations. Then we have an interesting issue with also ethnic differences. For example, we look at the moment at a particular mutation in the LRRK2 gene that occurs particularly in the European or in the Caucasian population. The same gene also shows mutations in the Indian population, but it's totally different mutations. And they lead to the same clinical outcome. So there's a lot of stuff that we simply do not understand.
Right. So we're still quite far away from understanding at a fundamental level what's happening in people.
Yeah, I think at the same time, we make progress on the fundamental level as well as on the applied level. So this is not going one by the other. So I think it goes all in parallel. Mm-hmm. And there is tremendous progress being made, but there's also lots of stuff that still needs to be done.
So what would you say one of the problems that we've had in trying to fully understand what's happening in Parkinson's disease? Would you say that it's the way that we've been researching Parkinson's so far, using animals and things like that? Because I know there's a lot of problems with translating what we find in animals into what's happening in humans.
Yeah, but for Parkinson, I think this is relatively clear that the animal models that we have are not very predictive for the actual human disease. There is certain things that up to date we can only do in animal models, and we don't have proper replacement. It's probably just a matter of time, but up to date we don't have them. But for really understanding particular disease processes, animal models are probably not very suitable. So that's one of the reasons why really time ago we switched to in vitro models, and then about 10 years ago to the organoid models, because they seem to be way more predictive.
And so this is what you were sort of looking at in your project that you've recently finished with us. You were growing your midbrain organoids. Could you tell us a little bit about sort of what you were doing with the funding that you had from us and what you found?
Yeah, probably first in a nutshell what midbrain organoids are. So these are kind of three-dimensional representations of the human midbrain. They're derived from patient-specific stem cells. They're about 2-3 millimeters in diameter and really nicely recapitulate the human midbrain in all its complexity. And in the funding that we recently received, what we looked at is trying to replace two components that are still animal-derived in the protocol when using these organoids. One is these organoids grow in an extracellular matrix, in a gel, if you will, which is often derived from animal components. So we replaced or tried to replace that with synthetic material. And the other one is animal-derived antibodies that we try to replace by recombinant antibodies. So really going for the analysis or protocol components away from animal components to recombinant or synthetic components.
So that's not necessarily a straightforward thing to do. It sounds to a layperson like me like, oh, it should just be easy. You should just pour it in from this bottle rather than from that bottle. And, it should all just magically work fine. And why wouldn't the cells like it? Because they're going to be in a more human-like environment. So it should just be easy. Not so much.
No, unfortunately not. Particularly when it comes to this extracellular matrix, the material that is animal-derived there that we were using now for decades is super complex. You would think it's just a gel, a few proteins, a bit of water, that's it. But it contains lots of factors, growth factors in a particular organization, and rebuilding that synthetically proves to be very, very complicated. Yeah. So we try to rebuild nature, which is really complicated and not easy. For the antibody, it's easier. There, it's fairly nicely possible to replace the animal-derived antibodies with recombinant ones.
So how did you get on with making this switch then? Did it work? Did it work partially? Not work at all?
So the antibody thing worked quite nicely. So there we are, I think, really at a stage where we can say we don't need the animal-derived antibodies anymore. We can take the recombinant. Brilliant. For the extracellular matrix, we haven't found any alternative that is as good as the animal-derived. So what we can do now is actually do the thing completely without matrix, which is also fine, but it doesn't recapitulate all aspects. It's like a smaller version of the organoid, which is still good for certain applications, but not for all. So I think it partially replaced, probably.
even though, that sounds like it might have been a bit disappointing, sort of doing the research, you didn't find something that worked, that's also still a really important outcome, because now you know that specific thing you tried doesn't work. And, you can tell others not to try that, and maybe they'll try something else. And it's just about trial and error to find what does work for the specific thing you're trying to do. So I think, that's still a really good outcome. And like you say, you also had the positive results from the antibodies. So, yeah.
I totally agree. And research is mainly about failures, right? I mean,
that's it, yeah.
From time to time, something works, but normally it does not.
Yeah, it's not easy. So back to your organoids, what can you use them for? Is it about sort of testing the drugs? Can you grow them for a long period of time to see, the kind of degeneration that we see in Parkinson's disease? Is that something that you can model using your organoids?
Yeah, the nice aspect of the midbrain organoids is that they really recapitulate pathology to a quite good extent. So we do see degeneration of dopaminergic neurons. We can grow them for up to 400 days. I mean, we could do longer, but at some point we stopped. You do see protein aggregation, like Lewy body-like pathology, which is something you normally do not see in cell culture models. We do see neuroinflammation. So we really recapitulate pathology. And we use that for multiple things. On the one hand, we test therapeutic approaches, small molecules, so drugs, but also other things like viruses, peptides, antisense oligonucleotides. So really different things. But we also use them, for example, for patient stratification, deriving midbrain organoids, for example, for multiple patients, and then investigate how different they look like if they come from different individuals, how differently phenotypes are expressed. We try also then to correlate that with clinical data and really see how predictive that can be.
Brilliant. And what is the kind of longer term goal for that then? Would this be something, that you would potentially, you work with pharmaceutical companies to try and get them to incorporate these models when they're designing drugs?
So with Organic Therapeutics, with our spin-off, we do precisely that. So our clients are biotech and pharma companies developing therapeutics against PD. And they're typically approaching us and ask us to test their therapeutics in these patient-specific models. So this is already happening now. I guess the long-term vision is really what I said initially, like being a precision medicine service provider. The situation is as I tried to lay out, somebody's newly diagnosed with PD. A sample is taken. The sample is taken into stem cells. Stem cells differentiate into midbrain organoids. The midbrain organoids are treated with all 10, 20 components or compounds that might be available, and then report to the medical doctor, for this patient, this compound is effective, so he should get that as a treatment. This is, from my point of view, really where it goes to. Cancer is to some extent already there, like typically a decade ahead. And I think we're going to get there for Parkinson's and other neurodegenerative disorders at some point.
In 10 years,
That's
maybe.
Incredible.
Maybe in 10 years. Who knows?
Wow.
Well, hard to say, right?
Amazing.
It's time projections in this business is very complicated.
Yeah. Yeah, absolutely. We won't hold you to that.
No, thank you.
But it would be good. Fingers crossed.
It would be good. What made you want to get involved in, I guess, Parkinson's, but being a researcher, being a scientist? Is it something that you always wanted to do, Jens?
That's a good question. I probably that just happened one step after the other. But it's obviously, it's a very nice career because it gives you so much freedom to follow curiosity, to basically, if you see something when you look through the microscope, and then the feeling or the knowledge that probably what you see there is you might be the first human on the planet who have ever seen that.
Oh,
that's
cool.
extremely cool. Yeah. And this is extremely fulfilling.
So if you had like one message for our supporters or for decision makers with, the ability to kind of influence the direction of research, what would you say to people?
That's a really good one. I think particularly for biotechnology or for biology, it's very important to work on a long horizon. So don't expect results really quickly. These things all take time. And if we talk about neurodegeneration, this is a process that takes very long. If you want to model it in the dish or in any other model, it also takes very long. So patience is probably quite helpful.
Yeah.
And don't be afraid of failure.
Well, thank you so much for the brilliant work that you're doing. Parkinson's is such a devastating disease. So anything that we can do to help to kind of, get closer to better treatments for people, we're very happy to do it.
Yes, I totally agree.
Thank you so much.
Thank you, Jens.
Oh, it's so interesting to hear more about Jens's work, Gemma, and it really gives me hope that brilliant people like him are, starting with the human when they're looking at Parkinson's disease, because that's clearly what we need to have happen.
It is. It's such important work. It affects so many people all over the world. So I think by taking it back to the human so we can understand what's happening is definitely the way to go.
Gives us hope for a brighter future ahead.
It does.
All right, Gemma, so what is the poll question for this week?
So the question is, do you think companies like Neuralink should stop testing brain devices on monkeys and focus on developing human-specific alternatives instead? Yes or no?
Very good.
So you can vote on LinkedIn, Facebook, Instagram, X, and Carla's Substack, the Animal Free Lab Stack, and we'll discuss the results in the next episode. And if you'd like to tell us your thoughts about the poll or anything else about the episode, then please leave us a message on 0208 054 96 95. That's 0208 054 96 95.
Gemma, tell us the poll question one more time.
It's, do you think companies like Neuralink should stop testing brain devices on monkeys and focus on developing human-specific alternatives instead? Yes or no?
Let's see what people come up with this time then. It's be interesting to see if we get any comments.
Yep.
So we're going to be back in a fortnight on the 25th of April, and we're going to be talking about World Day for Animals in Laboratories, which is the 24th of April. So it's an annual day where people mark the use of animals in laboratories and work towards ending them. So
yeah,
very relevant.
Very important day.
Okay, so thanks so much to listening, everybody. Please stay part of the conversation by reading my companion blog on Substack. You can just search for Animal Free Lab Stack and you can read and subscribe. But thank you so much for joining us today. Thanks for listening, and we'll see you next time.
Thanks for listening. See you soon.
You've been listening to the Animal Free Labcast, the podcast that puts humans, not animals, at the heart of medical research. If you want to find out about more ways to move to a world of animal-free research, then head to our website, animalfreeresearchuk.org. And if you'd like to send us an audio message about the poll or anything else you've heard in the podcast, then leave us a voicemail on 0208 054 9695. That's 0208 054 9695. Thank you for listening.